I want to start by thanking the organizers for the invitation and for organizing the meeting. I know it takes a lot of work to organize a meeting, so thank you very much. My title, Lipid Nanoparticle Mediated Delivery of CRISPR RNP Results in Widespread in Vivo Genome Editing in the Mouse Cornea. I'm going to start right away with my thank yous to the people who did the work. Zainab is a very talented graduate student in my laboratory. She was supported by Deanna, a research tech. And we did this in collaboration with a company called Incisive Genetics. This is a new startup in the Vancouver area. Austin Hill and Pamela Wagner helped with the experiments and were supervised and guided by Blair Levitt. So just to reiterate from the point of view of conflict of interest, this is a company-funded study. So definition of gene therapy. Gene therapy uses nucleic acids as reagents to alter cellular behavior and thereby cure disease. So we're pretty familiar with the three classes of therapeutics with the first version, small molecules like aspirin or Advil. Proteins are also a therapeutic class, typically insulin for diabetes. And then this newer class, nucleic acids. It could either be DNA or RNA in gene therapy. So just so you don't think it's like way in the distant future, there are currently ongoing 33 clinical trials for CRISPR-Cas9 gene therapy. And again, I'm also using the clinicaltrials.gov website for this data. A substantial number of them are in vivo, and of greatest interest to this group would be Leber's congenital amaurosis 10. A substantial number are ex vivo, and I'm going to be talking in my talk about both types of approaches. Everything I have to say is in mouse. The mouse that we're using is called the SAY mouse. Small I is what SAY stands for, and it's a mouse model of aniridia. It's a single base pair variant model, and the change creates a stop codon. So it's the most common type of mutation found in human patients who do carry a single base pair change, and it is also found actually in patients. If I show you the phenotype in humans, the first picture, just a normal eye. The middle picture, an eye with a hypoplastic iris. And then the third picture, sort of end stage with a cloudy cornea, and I think you can barely see, but the blood vessels growing through. Here's the mouse model. So in a normal mouse eye, the iris is larger and you don't see the white of their eyes. In the middle picture, you're seeing the enlarged pupil due to the underdevelopment of the iris. And on the right, you're seeing an end stage again of a cloudy cornea with blood vessels growing through it. So in my lab, the overall hypothesis is increasing the expression of PAX6 will improve the structure and function of the eye and ultimately rescue the disease phenotype. In the talk today, everything is the delivery of CRISPR. So CRISPR is just an enzyme, Cas9, that binds an RNA that guides it to the right place in the genome. And then in all the work I'm talking about today, we go down the pathway where DNA also is used to create the repair in one case or the damage in another. So let me say a little bit more about that. There's two big methods for delivering gene therapy. Adeno-associated virus, AAV, and lipid nanoparticles. Normally you hear me talk about the first. So at this meeting a year ago in Virginia, I was talking about AAV. But today is my first presentation about our work on lipid nanoparticles. Lipid nanoparticles, there's a long list to discuss, but for this talk, the most important thing is the cargo size flexibility. You're able, compared to virus, to put a large cargo in lipid nanoparticles. That's particularly important for us. My student's going to give a talk later in the day about the fact that we're now using the adenosine base editor, an even larger protein than Cas9. But the con is low transfection efficiency. And that's the real problem, and that's the problem being addressed in the work I'm going to present. So a little bit more detail about what we're delivering. The RNP is Cas9 combined with the guide RNA to direct it to the location in the genome. This is not a plasmid expressing Cas9. This is purified Cas9 protein, a template DNA, and they're encapsulated into a lipid nanoparticle, which is a double bilayer of lipid. And so the encapsulation process is done in advance, and you're delivering to the cells the encapsulated ribonucleoproteins, proteins, DNA, all inside of a lipid particle. Advantages are that the RNPs do not need to be transcribed or translated in the cell, so their impact is very rapid on the DNA. Second, they are degraded rather rapidly, and that's good too in a CRISPR situation where you may not want your enzyme cutting the DNA over and over again with time. My talk today really is in 4 parts. There's 2 sort of themes. The first is a reporter system. I'm going to talk to you about how we're using it, but it's a tomato reporter. The second is the correction of the base in the same mouse. In both cases, I'm going to start with ex vivo and move on to in vivo. So starting with tomato in cortical neurons ex vivo. This is the reporter mouse that we're using. It was designed for CRI, and many of you may know it for CRI. So it's a stop-lox tomato. In its native form in the genome of the mouse, there's no tomato expression. Then you come along with the CRI enzyme, causes a recombination between the lock sites, and now the stops are gone and you turn on tomato. You make a little extra DNA that just gets degraded. We wanted to co-opt this system and turn it into a CRISPR reporter. So the exact same mouse, but now we're going to add a CRISPR strategy and try to remove the stops and turn on tomato. In greater detail, the lock sites actually flank 3 stops, and then there's tomato. We're not the first to try this. There was a guide chosen that binds both locks in one publication, and another publication added an additional guide, so 2 guides in the literature. We tried these to turn on tomato, and we found them very inefficient. We added 2 more guides of our own, which would cut multiply through the stop region. And everything I'm going to talk about today involves using all 4 of those guides. But then we went one step further and added a template DNA designed to act as a band-aid and bring together the 2 pieces of DNA that we would like to have come together from lock site to lock site. Here we just ended up with 1 template as being sufficient, so 4 guides and a template in all the CRISPR reactions that I'm talking about for tomato. So what you hopefully can see, I know the room's a bit bright, is on the first column is a dye, hoist is a dye for nuclei. So everywhere you see blue, that's an individual cell. In the middle is the tomato. Those cluster of cells that you see there have turned on their tomato. At the bottom is a negative control. So you should see no tomato here, and then the blue and the red will merge on the right-hand side. So all my slides will be laid out very much in that way. So we did turn on tomato, but we found that we could turn it on much more efficiently if we treated the cells twice. So these cells are cortical neurons from embryonic day 17 and a half embryos that carry the tomato turned off in their genome, and then are turned on in culture when we deliver the nanoparticles. And you can see twofold brought it up to about 30% of the cortical neurons have rearranged their genome. Now I want to go through exactly the same kind of experiment, now talking about modifying the PAX6 gene. So this mouse that we make cortical neurons from is different. It carries SAY, as you can see here, but it is also a mouse we published a few years ago that we have added a flag tag to the 5' end of the protein. And so when the mouse has no CRISPR happening, this gene is dead because the SAY is a stop and the protein is degraded. When we do the CRISPR reaction, the SAY is corrected. Now we make a full-length protein which will be marked by the presence of flags, so we know that it's coming from this chromosome. So when we do that in culture, again, these are cortical neurons in culture. We use two different conditions here for the CRISPR reaction, and then the bottom row is our negative control. Hoist is the blue dots. Hopefully you can see something. I barely see them. For all the cells. These cortical neurons express PAX6 very nicely, and that's the green in the second column. And then the anti-flag is red in the third column. And you can see that the negative control is negative, but the two that got treated with CRISPR have corrected that base pair and turned on that protein. So we're really pleased with this, and I underline this mouse for people who are interested in nonsense suppression, for example. It would be the same system could be utilized. Here's some quantification of that. So of the PAX6 positive cells, we're getting 38% of them. So we're really pleased with that number where the correction has happened. We wanted to sequence and be sure they really were correcting. And what you're looking at here, these mice are heterozygous. So there you're seeing the two bases, both the T and the G. The T dominates just because of the sequencing reaction in the untreated mouse. But following that straight down, you can see in the treated cells, we're not talking mouse yet, they're mouse cells in culture, the G is now dominant because of all the correction that has taken place. And we can quantify that. So now I'm moving to in vivo. Our in vivo delivery for this study was intrastromal injection. This is literally injecting straight into the stromal layer of the cornea. This is used in humans for severe cases of infection to deliver antibiotics or antifungals. It also has been used to deliver pigmentation to the cornea. In mouse, things are very small, but this is still a doable approach. So what we're injecting into the cornea is our nanoparticles carrying our CRISPR. And the mouse is a mouse that has that tomato allele in its genome. The tomato is off in the mouse before we do the injection. And what you're looking at here again is blue marking the nuclei of the individual cells, red showing where a cell has received the nanoparticle and had its genome cut and turned on tomato. On the left is a SAY, we call it Phe because flag is in this mouse, and on the right is a wild type. If we zoom in, we're using a confocal microscope, The negative control is on the right. We can see that the cells of the cornea that are endothelial and stromal have been transduced. You might also see that you do not see transduction of the epithelial layer, and that will be important in a moment. So now let's do it in a PAX6 mouse, where you would expect to have flag turn on. But really, you wouldn't expect to have flag turn on. So the upper line here is our positive control. You're looking at corneas from an injected mouse, upper control, no injection, but it's a control mouse that expresses flag in its cornea. So only the epithelial layer in green expresses PAX6, and that expresses flag as well, as expected in the control. But because we know our nanoparticles didn't reach the epithelial layer, we can't expect to see flag in our injected corneas, and we didn't. Let me just do my summary slide. So this is the first LNP delivery of CRISPR RNPs and template in the eye. Never mind the cornea, in the eye at all. LNP CRISPR corrects at 38% ex vivo. And for the people who are interested in iPS cells and differentiating in vivo, ex vivo, etc., this would be something of interest. CRISPR delivery for tomato is widespread in the cornea. That's where you saw the tomato cells throughout the cornea. For some diseases, this is ready to go. So CRISPR may be immediately applicable to corneal diseases such as granular corneal dystrophy or Fuchs endothelial corneal dystrophy. But for aniridia, we need more work. So the 21 days is when we harvested after injection. And that may have been either too late, so perhaps we did transfect the epithelial layer, but it turned over already before we did our harvest, or maybe too soon, because we have to wait for those epithelial-like or stem cell-like cells to produce epithelial for us to see our rearrangement. So we will do more work on the timing of the harvest to really decide whether these particular LNPs can give us transduction of the epithelial layer. I said questions and comments, but I know we're saving them for later. Thank you.